This is my bacterial transformation slides with unit connections from AP Bio. So from Unit 1, I connected the structure of the GFP protein. It connects with topic 1.5, which is structure and function of biological macromolecules. Basically what 1.5 was about was that proteins have primary, secondary, tertiary, and quaternary structures, and their structures determine their function. In the pGLO lab, the GFP protein exhibits the secondary structure of a protein, and it has many alpha helices in the middle and then a beta barrel. It does look like a quaternary structure at first, but it's not because it only has one chain. So from Unit 2, I connected the lab with topic 2.4, which is about the plasma membrane. What that was about was basically the cell membranes consist of phospholipid molecules, which also are embedded with proteins, steroids, glycoproteins, and glycolipids. In the lab, when we treated the cell with calcium chloride and heat shocked it, we were able to make the plasma membrane more permeable so that the uptake of DNA is induced by creating pores in the plasmid cell wall. The heat shock allowed permeability so that we could allow for genetic transformation to occur within the cells. From Unit 3, which was cellular energenetics, I connected the lab with topic 3.6 of cellular respiration, which discussed fermentation. Fermentation is an anaerobic process that most prokaryotes go through that are anaerobic. They use energy from biological macromolecules in order to produce ATP. Specifically, lactic acid fermentation is what bacteria goes through to make pyruvate, which creates the byproduct lactate or lactic acid. In the lab, the bacteria uses fermentation when metabolizing the lactose that we add, or also the sugar in the agar, to produce a lactic acid to lower the pH of the environment and allow the bacteria to grow. Unit 4, which was cell communication and cell cycle, I connected topic 4.6 cell cycle with the lab. This topic discusses how cells divide to increase cell growth and DNA variability. So as the bacteria cells divide, they are going through binary fission during mitosis, and they're creating a colony or a lawn. This is seen in the GLO tray where the bacteria has grown into either a colony or a lawn. For example, in lane 1, it grew into a lawn, and then in lane 4, it grew into a colony. It divides through binary fission, which is specific to prokaryotes. Unit 5, which is heredity, I connected topic 5.1, meiosis and genetic diversity, with the lab. This topic discusses how prokaryotes only have one chromosome rather than eukaryotes. And they also contain a plasmid, and the plasmids are exchanged with other bacteria through conjugation. This connects with the lab because the bacterial cells, this conjugation allowed the bacterial cells to grow colonies in lane 4 because the pGLO plasmid was exchanged to other bacterial cells using conjugation. Unit 6, which was gene expression and regulation. Topic 6.7 discussed mutations, which was basically about how any changes in the genotype or any mutation can affect the phenotype, and then if this alteration of the phenotype benefits the organism, it will be passed along via genetic information to the offspring. So in the pGLO lab, the pGLO plasmid was artificially added to the bacterial cells, so it was basically like a mutation to the cells. However, this mutation benefit the bacterial cells because it allowed them to be resistant to the antibiotic, and survive. So they kept growing. Unit 7, Natural Selection. Topic 7.2, Variations in Population. Talked about antibiotic resistance and how if a organism it becomes resistant to a antibiotic, it will create variation within the population. In nature, usually, like if a virus becomes resistant, it will become a new strain. So in the lab, the bacteria's resistance to the ampicillin is due to the transmission of plasmids, which is the pGLO plasmid, which allowed it to be resistant. The passing of plasmid allowed the bacterial cell to adapt to a new environment, and the cells that can resist the ampicillin survived and reproduce, and the ones that didn't survive obviously didn't pass on their resistance to any offspring, and that's displayed by this picture. And lastly, Unit 8, Ecology, topic 8.7, which was disruptions to ecosystems, also kind of covers antibiotic resistance. So in topic 8.7, it discusses how an adaptation is a genetic variation that is favored by selection and manifested as a trait that provides an advantage to the organism. Usually when we think adaptations, we think of, like mammals and birds and that kind of thing, but we also see this in bacteria, specifically in the pGLO lab. The bacteria cells adapted to the antibiotic and were able to grow because they were at a genetic advantage.